So if you're preparing for Biology Paper 2, this is one of the important practical using quadrats. Please review this. The aim of this practical is to investigate how distribution and abundance of small organisms vary in an area using quadrat. The method is quite simple. First, place a 1 meter square quadrat on a ground at random points. Randomness can be achieved by using random number generator to pick the coordinate where you want to place the quadrat. That is very important point. And why do we do this? Because it helps you to obtain the results that are representative of the whole area. Okay. Now count all the organisms you are interested in within the quadrat. Repeat step 1 and 2 lots of times and work out the mean number of organisms per quadrat within that first sample area. After that, repeat 1 to 4 for the second area that you are comparing. Finally, compare the results. This is very important because students often write the whole method, but they forget to write that they must compare the results for the two sample areas. If this is not mentioned, you wouldn't get the full marks. Simple. Now, let's talk about the variables. The independent variable is the location being investigated. Remember, the independent variable is what you change. The dependent variable, on the other hand, is what you measure. In this practical, it is the number of organisms you're counting. The control variable, like size of the quadrats, area sample, time of the day, weather conditions, same observer, these should all be kept the same. Now, let's go over some common mistakes students make. Students often take too few samples or forget to calculate a mean, do not use random sampling. They count organisms outside the quadrat, use different size quadrat. They forget to state the variables. They forget to write the units. Counting organisms touching the border which is incorrect. And the rule is count organisms that are more than half inside the quadrat or exactly half inside the quadrat. Do not count any organism if it is less than half inside the quadrat. Now let's go over examiner must know points. Take at least 10 quadrat samples. Use random sampling to reduce bias. If an exam question asks why should quadrats be placed randomly, the answer is to reduce bias. Simple. Reliability can be improved by repeating measurements and calculating mean. Okay. If an exam question asks you how can you improve reliability, this is often 1 mark question they are looking for. Okay. Accuracy can be improved by using a large sample size. Please remember that. Finally, let's go over the calculation part because this is very important. First, calculate the mean number of organism, which is total number of organism divided by the total number of quadrats. Then take a picture of this whole calculation part because this comes up quite a lot in the exam. Next, calculate the number of quadrats that can fit into that habitat. To do this, calculate the area of that field, then calculate the area of the quadrat if it is not given, then divide both of them. And this will give you the number of quadrats. Okay. So finally, to calculate the population estimate, just do mean number that you, of the daisies or the organism, times it by the number of quadrats you've calculated. The mean is the value you calculate in the first step, and this number of quadrats is the value you calculate in the second step. And I hope this helped. Please do let me know if you have any question. There is a link attached below where you can access and download this revision sheet if you would like a copy. Thank you very much. I hope this helped. Thank you.